Okay, in this video, we are going to be looking at the process of the electron transport chain. Now, the electron transport chain is going to be occurring in the inner mitochondrial membrane. The majority of the components are associated with the membrane structure, and it's going to involve a series of redox reactions. Now, recall, the electrons are being held in the form of NADH. And the NADH was generated primarily in the TCA cycle in the mitochondrial matrix. Now, these electrons that are bound to NAD to form NADH provides the reducing power to drive the electron transport chain. So we are going to be performing a series of redox reactions, reduction oxidative reactions, along the chain in order to move electrons. It's called a chain primarily because we are going to be passing the electrons from one complex of proteins to another. The other term for the electron transport chain is oxidative phosphorylation. The term oxidative phosphorylation comes from the fact that we are going to be oxidizing NADH to NAD, and in doing so, we are going to be removing electrons and moving these electrons down the chain. So a series of oxidative reactions will occur to move the electrons down the chain, and ultimately we are going to be harnessing this energy, as we'll see in a second. The phosphorylation reaction is the last step of the electron transport chain, and this is where we are going to be converting ADP into ATP. So the phosphorylation of ADP using inorganic phosphate to generate our highly reactive energy molecule, ATP. Now let's take a close look at the components of the electron transport chain. Again, this process is going to occur in the inner mitochondrial membrane. So this is the inner membrane of the mitochondria. Now, the orientation is critical in this process, and so the mitochondrial matrix, I'm drawing it at the bottom, and the inner intermembrane space will be on the opposite side of the membrane. So the first chemical process of the electron transport chain is going to be taking the NAD that we generated from the TCA cycle reactions, and we are going to perform an oxidative reaction to extract the electrons that are being held on NAD. So this is going to be done by an enzyme complex that is embedded in the inner membrane of the mitochondria, and the NADH will be oxidized to NAD, and the electrons will be placed on this complex of proteins. So this first reaction is done by the proteins in the mitochondrial membrane, and these are called complex 1. The more formal name is NADH dehydrogenase. So this enzyme complex job is to extract the electrons from NADH and place them into the entry point of the electron transport chain, which is complex 1. Now, these electrons have to be held somewhere, and so the complex is going to have what are called iron-sulfur centers. These iron-sulfur centers are essentially a lattice work of alternating atoms of iron and sulfur, and the lattice, the iron, will be holding the electrons. So in this chemistry, we are oxidizing the NADH, and we are reducing the iron in this iron-sulfur complexes. Now, in doing so, this process, of course, of moving electrons is going to have energy associated with it. Now, this energy is going to be harnessed into performing work by the complex. So after we provide this redox reaction to move these electrons from NADH, providing NAD to continue the TCA cycle to generate more energy in the future, these electrons now have to move to the successive steps down the electron transport chain. So the next step is to move these electrons to a small molecule called CoQ. This is a small, lipid-soluble, hydrophobic molecule that can undergo redox reactions so that the iron-sulfur centers in complex 1 are oxidized. The electrons are then moved to reduce CoQ. This is also called coenzyme Q10. Now, in moving electrons, this is generating some energy. We are going to take advantage of the movement of these electrons, and we'll see the energy where it comes from and the description of it as voltage potential and redox potentials in our next video. But we're going to take advantage of the movement of these electrons to generate work. So energy converted into work. So the electrons themselves essentially have potential energy. The electrons now will move down an energy gradient as we move down the electron transport chain, and the energy that is released from moving down this energy gradient is going to be used for work. And what this work means is that we are going to pump protons from the matrix of the mitochondria into the intermembrane space. Now for complex 1, we are actually, the energy of moving 2 electrons will actually move 4 protons. So you can imagine how this would be done, is that the energy harnessed by moving these electrons in the redox reaction, that release of energy will cause a conformational change in complex 1 and take protons from the matrix and shift them to the intermembrane space. So on this very first step, we can actually see that we're separating protons and we're creating what is called an electrochemical gradient. The gradient is because we have a higher concentration of protons in the intermembrane space than we do in the matrix. It's an electrical gradient because we are having a separation of charge. This is a proton, so the protons have a positive charge. So we will essentially be creating a positive charge on the outside of the membrane and a little bit more negative charge on the inside. It is a chemical gradient because protons are chemical. And so we have a higher concentration of protons in the intermembrane space than we do in the matrix. Now, once the electrons are associated with CoQ, CoQ is a lipid-soluble molecule. So that means it can diffuse readily within the membrane itself. And so the job of CoQ is to just transfer the electrons from complex 1 to the next complex. The next complex that we encounter is called complex 3. Yes, not 2, but complex 3. This particular complex also again has redox reactions, so that now the electrons are moving to complex 3. Now notice here that we go from complex 1 to complex 3. So what about complex 2? Well, complex 2 is actually partially embedded in the membrane. Recall the enzyme that we talked about in the TCA cycle called succinate dehydrogenase. This enzyme was responsible for generating the double bond from succinate to make fumarate. And in doing so, we are performing a redox reaction, and we are generating FADH from FADH2. Now these are the two electrons that are extracted from the two carbons, the two central carbons of succinate to create that double bond. They are placed on FAD to generate FADH2. Now the question becomes, and we left this open on our video, is how do we regenerate the oxidized form of FADH2 in order to continue the activity of the enzyme in the TCA cycle? What we do is we place this enzyme within the mitochondrial membrane, and recall that the electrons that are bound to FADH2, this cofactor will be bound to the enzyme permanently. It's not like NADH that can be soluble in the matrix. So these electrons, during this oxidative reaction, these electrons are being transferred to CoQ. The reason this is called complex 2 is because when the electron transport chain was originally studied in a biochemistry lab, the individual complexes were isolated and then they were reassembled to see where the electrons go. So for example, if you take complex 1 and complex 3 and just take those two alone and put them in a membrane structure, you would not get electron transfer. You need CoQ to mediate the transfer of these electrons between complex 1 and 3. Likewise with complex 2. Complex 2 was purified and identified as part of this chain. And so it received this name, complex 2, but it actually is not part of the electron chain directly. It's indirectly associated through the TCA enzyme, succinate dehydrogenase. So in summary, complex 2 will take these electrons from FADH2 to oxidize it back to FAD to keep the enzyme going, and these electrons will be moved to CoQ. Once they're in CoQ, then they can be transferred to complex 3. Now likewise, with the complex 3, we need to move these electrons along, and so these electrons are going to be moved to another small molecule called cytochrome C. Cytochrome C actually has heme iron bound to it. And the difference between this heme iron and the heme iron that we saw in hemoglobin is that this heme iron is in an oxidized state. So the iron is in a ferric state, 3+. This being the case, it can accept electrons, and we are going to be moving the iron from the ferric state to the ferrous 2+ state. And this is how cytochrome C is able to handle the electrons coming from complex 3. Now cytochrome C is going to serve the same function as CoQ. It's going to transfer electrons to the next complex in the electron transport chain. This complex is called complex 4. Complex 4 also has a formal name that is called cytochrome oxidase. Now as the electrons, let's back up here to complex 3, as they are removed, as they are oxidized and removed and placed on the cytochrome C, again it's coming down an energy gradient in the same manner as complex 1. We are going to harness this energy in complex 3, and in this case we are going to pump protons again from the matrix of the mitochondria to the intermitochondrial space. In this case, we are going to be pumping 2 protons using this complex 3 structure. Now, once we arrive at complex 4, after cytochrome C is oxidized to bring the iron back to its ferric state and release the electrons onto complex 4, this is the final stage of the electron transport chain. And we are going to now move these electrons onto the final electron acceptor in the energy metabolic process. And this is going to be, of course, oxygen. In this process, we are moving the electrons onto oxygen, and we are going to generate water from this process. So we are going to need 2 protons. The electrons will come from the complex 4 cytochrome oxidase, and we will now reduce oxygen to form water. This is a transfer of electrons, of course, and so again we're going down an energy gradient. Complex 4 will use this once again to pump more protons, to expand the proton gradient from the intermembrane space to the matrix. Now, once the electrons are associated with oxygen to form water, the electron transport chain is complete. Now, we have to concern ourselves with this proton gradient. We are now going to use this proton gradient that we have established to perform work. So the overall plan of the electron transport chain is to use the energy drops in electron movement from complex 1 to complex 4 to continuously pump protons and create a gradient. The gradient can be considered potential energy. So we are storing the energy from our oxidation of glucose into NADH, and then we are taking this energy and we're using this energy to transform it and store it in potential energy in an electrochemical gradient of protons. Now, the final complex that we need to concern ourselves with in the mitochondrial membrane is what is called the ATP synthase complex. This is the final enzyme structure that will be taking ADP and converting this to ATP. Now, obviously, this is creating a high energy bond. So the conversion of ADP to ATP is a positive delta G. This is an unfavorable reaction, so we have to put energy into the system in order to generate this ATP. The energy that's going to be used is going to come from the gradient. So as the gradient is allowed to dissipate through this complex of ATP synthase, this movement of the gradient, we are losing potential energy by dissipating the gradient. And what we are going to do is turn it into kinetic energy. Kinetic energy means that we are going to be spinning what is called the head group of the ATP synthase complex. So this is actually what is called a molecular motor. As this spins, the kinetic energy is going to be harnessed to generate ATP. And we'll take a look at this process when we look at redox potentials in the next video. So the dissipation of this gradient, proton gradient, we are releasing potential energy, converting it into kinetic energy, and the spinning of this molecular motor is going to generate the energy to put into this creation of this high energy bond to make ATP. So that will take the unfavorable reaction and make it a favorable reaction.